So I will be actually presenting, the topic will be pluripotent stem cells, derived approaches for regenerative medicine and disease modeling. So it will be actually a bit more based on regenerative medicine and cell therapies. And basically, I think it's very, very interesting that there are more and more evidences that there are differences about different species. And actually, most of our research traditionally has been actually based on animal models to try to understand disease. And now we think that perhaps actually we were not tackling the questions in the best way. We were skipping maybe some very important information that actually we can only achieve by the generation of new tools that they are based on human research, let's say. So I think for that, it's tremendously important to generate stem cell-based approaches, to generate better human models to tackle the two major questions. So there will be a study disease and therefore also find more effective therapies. So also on the other hand, there are also increasing amount of clinical therapies right now that they are using stem cell-based approaches. So basically, they're trying to generate different tissues and different tissue cell types for transplantation on patients. And I think for that, human pluripotent stem cells is a very attractive tool because it brings to the market a very scalable source of cell types. And also is very versatile because, of course, from the same cell type, you can generate many different tissues. And that can be used for transplantation for cardiomyocyte cells for the heart. It could be also the generation of pancreatic cells. As it is actually currently being used and addressed, it could also generate retinal pigment cells. And this is a major clinical trials going on, especially in Asia, so in Japan and in China. And it could be also a very interesting source for the generation of different brain cell types. And this is a little bit more my domain. Of course, we are a little bit far from there, but there are also a number of clinical trials that are also ongoing for the generation of, for instance, dopaminergic neurons for transplantation for Parkinson's disease. So I think we have the knowledge, what is lacking, or we are actually investing most of our resources and energy is to try to generate the right tools to really differentiate the specific cell type that we need for those transplantations and to have better quality controls for the cell products for the market.